and I'm happy to introduce Dr. Richard Judson from US EPA, National Center of Computational Toxicology. Richard has training background in chemistry, actually, and bioinformatics, and spent a lot of time with the consulting business and also with small biotech and pharma companies, and has been at the EPA for 3 years now, leading their efforts on cataloging a lot of data and also putting all of the new data that NCCT is collecting into databases. So we're very fortunate that Richard agreed today to give us a layout of the landscape of all the legacy and new data that's coming out, not only at EPA, but also in other government agencies that are collecting data.
Thank you, Vaughn, and the other organizers for giving me a chance to talk. Okay, but before I get started, I want to recognize that a lot of people view databases as kind of ugly children or ugly babies. They're things that only their parents can really appreciate. But I want to make the case that these databases really are important because they provide a lot of the glue for the other things we're talking about. So, you know, what we're trying to do here in computational toxicology is really start thinking about building models where we take tissue dose of some chemical, the chemical hits some sort of molecular targets, it causes cellular changes, network changes, and ultimately, potentially, whole animal toxicity. And there are a whole series of modeling efforts you need to do to be able to stitch all of that together. And the databases are an underpinning for a lot of that. Okay, the first set of talks we heard this morning, they didn't look very computational to me, and so I want to actually put some of this into context a little bit. So we're talking about computational toxicology, so where does computational come in? So we have lots of data. So Chris Austin was talking about generating data on hundreds of thousands of compounds, and the other groups are, again, generating lots and lots of high throughput, ultra high throughput data. And so you need to put that someplace. And so you put it into a database. And most importantly, you also have data workflows, because if you have to do all this manually, you just have a big mess. We have to organize the data in some way so you can actually ask questions of it. So, you know, somehow the data just doesn't speak to you all by itself. You actually have to get it organized and actually think about having question workflows as part of that. An important word here I'm going to use is not only databases, but also knowledge bases. I'll talk a little bit about what that is. If you build a big database that you want people to use, you'd better have it be web accessible, which has been an issue so far. You have to annotate data, and so a lot of this data doesn't mean anything if it all sits by itself. So you have to have databases linking to one another in some way. And another issue I'm going to talk about are these things called ontologies, and I'll tell you what that is if you don't already know. It's another important way to annotate data. Once you have all the data in place, you need to be able to do modeling, and I broke that into two levels. One are what I call simple statistical analyses, is hitting a particular receptor correlated with a particular endpoint. So you can think about doing t-tests and chi-squared tests and things everybody knows how to do. Or you start building high-level models where we think about networks and doing simulation and so on. We'll hear lots more about that later on today. So these are all the sort of things that we think about as being the computational piece of computational toxicology. It starts with having lots of data, though. Okay, so what is data that we can think about? The obvious thing are numbers. We have Lowell's, we have IC50s, we have concentrations, we have, you know, weights of animals, who knows what. You have parsable text. I actually would say that all of the 20 million or whatever PubMed abstracts that are out there are parsable. We bring them in. Lots of other people have downloaded all of PubMed, all of the abstracts, and you start building information or pulling information out of that. And so that's a kind of data. Structured relationships. And so, you know, this is a very bold statement that obviously needs lots of qualifiers, and I'll actually come back to that. You know, so you have these things which are subjects, objects, verbs, and so on, that really those concepts come together, and they're kind of data. Images are data. We've already seen some images today. And then there's kinds of metadata. So you do an animal experiment where you put some pesticide into some rats, and you finally, at the end of 2 years, you say, okay, did I get tumors or not? Well, that's the low L you get out of that may be a piece of data, but what strain of rat? That's a kind of metadata. How many rats per sex per dose group, for instance? Those are kind of metadata. If you haven't stored that sort of thing, then the data is essentially worthless. Okay, so you have to worry about that. Okay, what is not data? A lot of the world's toxicology data is still sitting out there in PDFs and TIFFs and GIFs. And occasionally somebody will come to me saying, oh, I have a database of information on some set of chemicals, and say, well, can you email it to me? Well, no, I can't, because it's this stack of papers in my file drawer that I Xeroxed from somebody else a few years ago. So there's probably still the majority of the toxicology data out there is sitting in these things that is not really data yet. There are other cases where you actually have data online, but it's in a tabular form in a report that hasn't really been parsed yet, so you can't really use it for computing. Okay, so now there's all this data out there. How do we think about pulling it together? And obviously don't need to read all of these things, but the sort of data that's really relevant to computational toxicology are chemicals, and there are big databases of chemicals and chemical structures. So PubChem is a huge, very important resource. DSS Talks I'll talk about a little bit. In vitro data, so the sort of data Chris Austin and the other two speakers this morning were talking about, kind of screening data. PubChem is grabbing a lot of those. There are a couple of other databases that are out there that we use. Tox Miner is our internal database. In vivo data, so all of the rodent data, other species data that we actually want to compare against and build models around. National Toxicology Program obviously has pulled a lot of that in, and various other databases that are out there. There's a lot of reference biological data, and anybody who's a biologist knows all of these, all of the, entree gene, entree protein, everything else that entree has put together. There's databases of ontologies, databases of diseases, pathways, literature, and so on. And then there are databases that are linking things together. Carolyn Mattingly will talk after I am about a very important link, what I call a linkage database. She may disagree, called Comparative Toxicogenomics Database. There's a similar sort of effort going on in Europe called Go3R. We have a virtual tissue knowledge base, which is doing some of the same things. Issues with some of these kinds of data is the data is in many formats. I'm going to come back to this. This is really the big issue right now still, is data is spread all over. It's in many different formats. It's hard to access. Some of it is impossible to access today. And much of the in vivo data, which we, in a sense, what we want to do is sort of move away from doing in vivo experiments for lots of reasons, because you don't want to kill animals if you don't have to, if you're not learning anything from it, but also you can't afford to do that. But most of the data, I would say actually most of the data, the good in vivo data, is still out there in a way that has not been parsed and is directly usable. Okay? Okay, but why bother? This is kind of obvious. All of that data, though, people have generated, it costs many billions of dollars to generate. And of course, we could never afford to rebuild that, and it would be evil, sinful, whatever, to kill a bunch more animals just because we didn't parse out the data that's already there. Okay, and predictive biology or predictive toxicology requires integrating over all of these different link scales, different kinds of data. We don't necessarily know how to do that today, but without the data, you obviously can't even get started. Okay, I'm going to show this slide again at the end. I'll bring it up and leave it at the back, but there are a series of very big comprehensive databases, and these are some of the ones I will touch on today. Okay, first start talking about the EPA actor database, one we put together. This was prompted, the development of this was prompted by a question that was posed when I showed up at the EPA about 3 years ago, was what are the chemicals that the EPA cares about? So this was my EPA colleagues asking me, is there a comprehensive set of chemicals that we really need to worry about as we go into this high throughput screening sort of area? And what do we know about? What does the world know about that? And those were unanswerable questions because these lists of chemicals and the data about them were spread all over. And so we started putting together a very comprehensive database, which is essentially initially just an index of all of the databases having to do with environmental chemicals that are out there. And today we're up to about 300, well over 350 essentially sub-databases that we've pulled in and indexed, about 500,000 chemicals. You can go search this. Just the sort of the bottom line of that initial query that we asked was, it turns out the EPA, the high priority chemicals at the EPA is about 10,000. Okay, so it's a lot of chemicals, although in Chris Austin's world, that's not a lot of chemicals. You can essentially screen all of those if we could actually get them into 96 or 384 or 1536 well plates, and you could screen all of them for a lot of things in a short period of time. But of that 10,000 chemicals, so these are things that people may be exposed to, only about a quarter of those have ever been tested in animals for carcinogenicity and reproductive toxicology and developmental and so on. Okay, and so there's a big data gap, which is, again, a big reason why computational toxicology is looked on as being really important, because it's a way to move away from trying to fill in that data gap, which we'll never be able to do with animals. Okay, ACTR brings together a lot of information on chemicals, on assays, and an assay is a very generic term. So is it binding to a particular receptor? Is it doing some sort of a whole animal 2-year study? It doesn't really matter. It all goes into these assay tables. All of the annotation data from the public databases on genes and pathways and diseases and so on. Then there's a series of workflows. Again, you can't do this by hand. So there is the middle workflow, simply taking all of the in vitro data that we and our colleagues generate, and how do you clean that up and make sure it's relatively high quality before you actually put it in the database to make questions about it. I'm going to come back to this more. What chemical did you actually test? Turns out that's a tough question to answer sometimes. And then once you finally have the data, how do you actually go through and ask all of the hundreds or thousands or millions of questions that you could actually ask given this data? And then the core ACTR database is tied into several other databases we have at the EPA. Okay, so this is a little eye test. This is the current set of 379 databases we've pulled into ACTR, which is in the release that's going to hopefully go live. ACTR is already live, but this is a new version that's coming out hopefully in the next week or so. And I'm just going to make a few points. Take that word tabular there and strike that. So there are few sources of primary toxicology data, and that's mainly the EPA, the Office of Pesticides, and the NTP, and there are a few others. There's lots of other auxiliary data, but most of that data initially anyway was not tabular. You couldn't directly go compute on that. There are a whole series, and I haven't marked all of these, of primary or secondary text reports. So IRIS, for instance, is kind of a secondary textual report about chemicals, which is not directly computable. There are a number of in vitro data sets out there, and again, there are more than I've highlighted there. But then there are efforts going on both at the EPA and other places to take these primary data sets and extract them into tabular format. And in particular, the DSS Talks program at the EPA is doing that. So to take this stuff, which is useful one chemical at a time if you want to understand that chemical, but if you want to look across chemicals and do some sort of computational modeling, is not terribly useful today. Okay, the DSS Talks project at the EPA was initially started to do this effort to essentially take databases or take lists of text reports, pull out tabular data that you could compute with, but also it's dealing with this very important problem. What was the chemical you actually did that study on? So if you go to the NTP study reports, those are actually very well annotated. They'll actually tell you something about the analytical chemistry on the actual compound that went into the test. A lot of the other, if you go into just a random published article, it may be difficult to tell, okay, what was the structure of the compound? What was the purity of the compound that was actually run? And without that, it's hard to pull data together and do various kinds of important meta-analyses. And so that's really the bottom line of what the DSS Talks program has been doing. And so there's a lot of detailed information in the DSS Talks database about what the compound is. What actually did you test? What was the test substance versus what's sort of the idealized compound that you would like to think you were testing? And for instance, there was a question about mixtures earlier. Okay, a lot of times you actually test mixtures, and if you have a defined mixture, okay, it was 50% that, 50% this, okay, the test substance won't actually have a chemical structure. It'll have that definition, that it was 50% this, 50% that, and then you can go back and say, okay, what were the structures of the things you actually mixed together, you bought from Sigma and put in there. There's a complicated, very comprehensive workflow to do quality control on the data to say, okay, really what was that chemical? And I won't say anything more about the details other than this has taken a long time to understand all the problems that people can throw at you and to develop better ways to clean it up and be pretty sure of what it is that you're actually, you actually tested. Okay, so here's a new concept. I'm switching gears here a little bit is something called ontologies. And an ontology is really a collection of things and relationships between things. Okay, and so I'll come back here to this little example. So arsenic is a thing, a bladder tumor is a thing, and causes is a relationship. And so one of the problems with exchanging data, pulling data together from many different sources, is that people can call the same histopathological finding two different things. Okay, and so what you really need to do is make sure that there's some way to take those synonyms and merge them together so that if somebody calls it A and somebody calls it B, okay, it really is, we'll merge those together and it's A prime, whatever it is. Okay, and so that's what ontologies do. They talk about hierarchy, so you can talk about disease, cancer, bladder cancer, and so on. And by organizing it into these concepts and hierarchies, that actually facilitates doing relationship discovery. Okay, so for instance, well, I'll get back to that in a second. So the basic concepts are individual. So this is arsenic and bladder tumors are two individual concepts. They're classes of things. So these are classes in the class of chemical and disease. They're attributes. So a chemical can have a molecular weight, a tumor may have a size, some sort of a size thing. And then you can put together these relationships. They're also modifiers, and they're, of course, very important modifiers for this statement to really be true. So arsenic causes bladder tumors in humans, so it doesn't do it in rodents, is my understanding. It causes bladder tumors in humans at relatively high doses, above some high dose, for very chronic exposure. And so we need to be able to build these relationships. But what's nice is if you can go into a database and query the database to look for evidence to support these sort of statements, it's much more powerful than giving somebody a p-value. It's really a statement of, maybe it's not a statement of fact, but it's a hypothesis that you can then go and look at more carefully. Okay, there's a huge effort, across the scientific community in developing ontologies, and so OBO, if you go to OBO Foundry, the Open Biomedical Ontologies database, there are hundreds of these. Okay, so any field where it makes sense to build an ontology, somebody has done that. And it's mainly in the area of diseases and genes and other biomedical concepts. So, development, how does an animal develop? What are the words you use in describing developing animals? Okay, switching back to databases, so ToxRef DB is a database we've developed again at the NCCT, and what this is doing is capturing data from guideline animal studies. Initially, it was just the Office of Pesticides at the EPA, but we're bringing in NTP and other high-quality data sets. It's a relational database, so everything is taken out of these text reports, put in a relational format, and it's really the in vivo anchor for the predictions from the ToxCast program, which I'm not going to talk too much about. There are several different study types and lots of different kinds of analyses we can do. This is a really hard thing to do, and it's not just us. Taking all of the toxicology data from the literature, wherever it is, and putting it into some sort of a computer form is a big deal. So on the left-hand side, you can't read this, of course. This is just a big block of yucky text. And so we have students who actually sit there and read that and put it into there's some forms that they have to fill into the database. And why I talked about ontologies is because all of these reports were generated over decades, and so the whole terminology has changed. And so they had to actually come up with, for each of the study types, a particular ontology and map all of the concepts onto that so that we could actually compare things from the 80s to the 90s to the aughts and so on. And these are just some examples of, this is a an animal that obviously has several developmental problems, and so you can actually then say that, okay, it has some sort of a general problem. There's a problem with the crania, the problem with the eye, a problem with the face, and those, exactly what they are, map onto these very defined codes from one of the ontology databases. ToxRef is, again, we've taken all of this data from text reports, we've mapped it onto a database so it's searchable. This is a user interface that's going to be online hopefully by the end of the year, where you'll be able to look into all of this data we've pulled in and build reports. And this is a report where we're looking for thyroid gland effects across a series of chemicals, and it gives some essentially what are a funny kind of low L's, and it tells you what species and strain it was done in. Okay, I'm going to come back to quality control again. So you have to be very careful what chemicals you looked at. You have to be very careful what it is actually was in the study you did. And are these students who were reading these yucky text reports and mapping them onto these input fields, have they done it right? And so we actually developed a series of SOPs. They do a primary data entry. If there's new words that we haven't seen before, it's not in the ontology, we have to go in and update the ontology. We have a secondary data reviewers, because this was pesticide registry data that came from companies. They obviously had a stake in making sure it was right. And so we gave all of the companies who had submitted the data initially the ability to go and review that. So this is the stakeholder review. And about 50% of the studies they made the choice to review, and the accuracy has been very high. Okay, so ToxRef is taking these guideline studies. The obvious other very important place where there's a lot of guideline, very high quality data on in vivo studies is at NTP. I just have one slide here. Right now, if you go to NTP, you get these big blocks of text that are not, you can't really compute with them, but there's an active effort there to essentially move the whole database into a big XML dump, which then we and whoever else can download and then do cross-chemical, cross-study comparisons. That's a really important project at NTP. There's another database, and I'm, again, just one slide to mention this. At the FDA, National Center for Toxicology resource, they have their liver toxicology, toxicity knowledge base, where what they're doing is they're going to the open literature. So they can't go to the studies that the pharma companies have given to them and pull the data out and publish it like we've been able to with the pesticide reports. But so they're going to the literature. But what they're focusing on are drugs, which are obviously very important. There are a lot of liver toxicity is a common phenotype. So that's going to be an important and interesting database also. Okay, so at the EPA, what we do is we initially have all of this in vivo data we are getting through ToxRef DB, and the in vitro data that we get from Chris Austin's shop and various other places. We need to pull that together and make sense out of that, and that's going through a database called Tox Miner, where we're merging in vitro and in vivo. It's a database that sits on top of the actor database you saw before. There's a lot of data QC. We're linking in external annotations from genes, pathways, diseases, and so on. And really the purpose there is you have all this data together. Can you start doing statistical associations, trying to discover relationships? And there's a big statistical engine, which is, there's nothing magical about it, except that it's tied to all of this interesting data. And what we're doing is correlating data from cellular assays, biochemical assays, hopefully genomics, we're not quite there yet, physicochemical data. We have in silico predictions. We have all the in vivo toxicology. And so we want to be able to take all of that very complicated, structurally complicated to think about data and discover things. And this is just an example. There's a whole talk behind this very fancy picture here. But what this is looking at is looking at progression of liver disease. So chemicals can do nothing to the liver. They can cause some sort of lesion, they can cause a pre-neoplastic lesion, or they can finally cause liver tumors, neoplastic. And these little yellow circles are genes that are associated with assays, and these links simply say this gene is associated, if you hit this gene or upregulate this gene, it's associated with this sort of endpoint. And what we see is that there are a lot of ways to cause liver lesions sort of general liver lesions that don't move towards pre-neoplastic or neoplastic, and these are all the xenobiotic metabolizing enzymes. And so, it's quite obvious if you just simply have some chemical that's hitting the liver, causing xenobiotic metabolizing enzymes to be upregulated, you're going to cause the liver to be a little unhappy, but that doesn't lead you to these sort of very bad endpoints. There are only a few ways to get here, and again, these are rodent endpoints. So PPAR gamma, PPAR alpha, this is another PPAR surrogate, the androgen receptor. Okay, and so there are a few ways that we're seeing to get there. And so it's kind of an emerging story of what we can do to make predictions. Okay, so I'm going to talk just a little bit about a knowledge base. A knowledge base is a new kind of thing where you start with data, all this data in the database, which is facts and concepts, and you're really adding the relationships. So you're adding the ontology sort of data. And then from that, you can actually look for these common patterns where you can discover the new relationships, like A causes B under the conditions C, D, E, and F. And so you obviously have to have the structured data and the ontology there. We're building a database called the Virtual Tissue Knowledge Base, and it has a pretty complex workflow where you start off with some keywords. Okay, what are the key concepts? The keywords are list of chemicals, list of diseases, lists of histopathology findings. You then pull in all of Medline, all of PubChem, and you parse all of that, and then there's this @node is a knowledge base engine that we pulled in from someplace else, and that's been embedded. And from that, you finally get this knowledge base web search tool. Right now it's an internal web search tool, but it'll be made available outside. So you can go start asking questions of this knowledge base then. And just an illustration of there's a lot of complicated stuff built in there, but it's mostly being built off of public data, public tools. And so, it's finding some interesting things about it, and but I'm not sure that we're quite ready to say exactly what we found. Okay, but it has some pretty pictures. Okay, so that's really my overview and future directions and the issues that I see. Okay, we need to take more high-quality toxicology data and move it into databases and knowledge bases, because this is really going to make that very expensive data useful for other things than it was initially used for, and we'll learn a lot more from that. And this is just hard work, and so we need, hopefully there are more groups than us who are doing this and doing it in a way that can be shared. We need to improve and expand the literature mining efforts. Obviously, the literature is a subset of all of that. We need to develop better infrastructure to allow reuse and linking of these existing databases. This is, one of the primary purposes of the ACTOR database is simply to say, okay, where is all the data? We need to be able to pull it all together and make it useful to other people. And we've actually uncovered things. There are a number of groups who've downloaded the entire ACTOR database we've developed. We'll just hand you the whole database, and then they can go do data mining on that. We have to further incorporate ontology information into the databases, because that really adds value. And we have to build better methods for predictive modeling, which is actually what we're going to hear about this afternoon. And finally, just to close, this is the list of data sources that I talked about here. Most of these are online. You can use the URLs, and I'm assuming all these presentations are going to be posted online.